Okay, can everyone see the slides?
Yes, sir.
So let us start our class today. And before we start our class, let us briefly look into and have a short discussion like what we have studied in the last class. So in the last class, we were looking into what are the techniques of means the preservation of the isolated strains. Like once we have isolated your target strain, how we can carry out the preservation techniques and like we have looked into like what are the major culture collection centers that are there across the world. Okay, so uh, can anyone tell like what are the means what is the culture collection center that are present in India, for example? Can anyone tell the two names for that? Hmm. We have already studied those things. Anand Keshav,
Yes, sir.
can you tell the two names of the culture centers that are in India? Anyone else? Is already written. Yeah, if you cannot see and tell the answer also, then it is uh, how do you expect that? Anyone else?
MTCC in Chandigarh and NCCS in Pune.
Yes. So there has been these two culture centers that are being that are there in India, where whenever suppose some new microorganisms is being isolated, they are being deposited at these two centers. Okay, so like this means, but when you are depositing your culture, there are different techniques by which the preservation of these strains takes place. So it can be in the way of by keeping the metabolic activity to be in the active state, or we have the suspended metabolic activity. Okay, so the metabolic activity of the microorganisms such as the growth, their cell division and all those things are stopped. Okay, so the two for the two different things we have seen what are the two different ways. There are two different approaches to maintain the to suspend the metabolic activity and to maintain the metabolic activity in such a way that we can preserve the stains, right? So we have seen that under this particular category, there's something like periodic transfer of the fresh media. We overlay the cells with the mineral oil, or we use some saline suspensions or with the sterile soil and so on. And in the case of the second case, we go for the drying of the cells in the vacuum and the lyophilizations, use of liquid nitrogens and storage of the silica gel. So, and why the preservation is necessary? So we have seen that it helps to protect against the contaminations. We eliminate any genetic changes that is there and retain the viability. So this because if you are for example going to use a particular microbe in an industry and you're going for a production of any particular biochemicals, we have seen that we would look to have a higher yield and the highest quality, and we might need to maintain that during the fermentation process. Okay, so whenever you are getting the stain, you should be of means it should have a genetically stable, and that it should not undergo any genetic change, because we know that microbes whenever they are means they are undergoing divisions through cell divisions, or they undergo mutations, then there is a change in the genetics structure, and that may bring into the changes in, for example, how much the product is produced, how much the yield and the quality of the product, and all those changes are variations can be come into action. And generally when you are using any industrial system, we would not like to have such type of variations. Okay, So for that, it is very important that we have the preservation of this type of microbes. And then we looked that this, for example, if you want to maintain the viability of the cells, that is, you want to maintain the metabolic activity means that can be done by changing the for example, the growth media components, that is by limiting the carbon, nitrogen energy sources and maintaining the temperature, preventing dehydration and so on. So generally there are short term and the long term methods, and under that we have seen that, for example, what is a basic protocol if you are suppose going to preserve to make a particular. So generally when you want to, for example, preserve the strains, we have this type of closed, completely closed and the packed, sealed ampules. So these small glass tubes are called the ampules, in which we have on the top, we put the cotton plug, okay, to prevent any contaminations to come in, as we, for example, we carry out the sealing of the tube. Okay, and below this we have generally the cell cultures in the freeze dried powdered form. So generally it is, so when we freeze dry any suspension of the cells, what we obtain is is a powdered. So we have the viable microbes in the powdered form. Okay, and that is being placed at the bottom of this ampule, and the top of the ampule you can see that a cotton plug is there, and that particular cotton plug you can see there is a burning at the top. Okay, so means when we insert insert a cotton plug, we heat it at one end to make sure that all any microorganisms that is there, suppose any contamination is there, that can be removed, and then we carry out the complete sealing of this top end of the glass vial, so that nothing can pass through it and across the these type of cotton wool and then to the microbes. So you try to maintain the complete sterility. Okay, and on that, once you have means you have preserved your microorganisms in this form of pellet, on the top you need to label the name. Okay, and that labeling should consist of, for example, the date, okay, of labeling, and it should contain the information. So like here it is written LMZ 19534. So we have seen that it can be LMZ represents a particular for example, the culture collection centers. So generally if you see, generally we have these ATCC, or the, in the case of Indian system, we have the MTCC. And when you get the microbial this type of these type of vials, it will be written some MTCC, some number. Okay, so it will, the MPCC will give you the information of the which, from which collection centers you have you have obtained your this culture, and the number 3126 will actually give you for example, the information about the microbial stains. And for example, in each and every collection centers have their databases on means of the informations on their websites, and they contain lot of information. So if you can if you go into those websites, they will contain information starting from the genus name, the species name, the everything details of the particular algorithms, what is the media components, what are the means application of these microbes, what type of biochemicals it produces, and so on. So it contains all those details informations in their databases, and each and every cell banks have their own database. Okay, then we have looked into we started looking and discussing on for example, like how what are the techniques by which we can preserve the microorganisms, as well as we maintain the metabolic active state. Okay, so one of them is the regular subculture, and generally if you see in the means subculturing in the liquid media is something that that we have to do for a long period of time, and generally it is prone to contamination. So so whenever you are doing this type of subculturing, you might need to do the culturing within, for example, every 7 days, or every maybe 3 days or 5 days, depending upon the type of microorganisms you have to subculture. Okay, but in the case, so that actually leads to, when you have the number of subculturings are higher, so that leads to the high prone, or high, I mean, it's a higher, it leads to the higher chance of, for example, the contamination. So to overcome that issue, we generally use solid media. And in the case of the solid media, we have plus. In the case of the solid media, we have, for example, we inoculate the cells onto the solid agar. Okay, and depending upon the type of the bacteria or microorganisms we have, either aerobic or an anaerobic, we can selectively choose like which type of cultures we will go for. Like either we will go for a slope culture or we will go for the step culture. So can anyone tell that if it is a anaerobic bacteria, which type of means solid media culture will you go for? Will you go for the slope culture or the step culture? Hmm. Abhiraj, Abhiraj, Abhiraj is not there anyway. So Abhiraj is absent. Amisha, Chinmay, Chinmay. See, everyone has to respond. So anyway, if you are not responding, then obviously I am noting down, and everyone knows, right, that you have a interaction marks of 10 marks for that. Okay, so if I don't get any response, so you have to respond at least. Even if you're saying wrong things, it's you have to respond. If there is no response, then uh, I will obviously directly put zero on that. Uh, Rituraj,
Yes, sir.
can you tell the answer? Like we have the slope culture and the step culture, for example. So if you are going to make a bacteria, means if you have a aerobic system in which or anaerobic system, for example, we will go for the which type of slope or the step type of culture. Rituraj, you know the answer? Shahil,
Yes, sir.
do you know?
I think for sir, for aerobic bacteria we will use step culture. For an anaerobic we will use slope.
Can you repeat? For aerobic, you will go for the
Step
step one.
culture. Yes, sir.
No, it is a reverse. So for the anaerobic, we will go for the. So anaerobic is what? Anaerobic are the organisms which doesn't require oxygen. Okay, so they will grow in the means absence of oxygen, and that's why. So in the case of the anaerobic, okay, we do not require any oxygen. Okay, so oxygen is not required. In case of aerobic, we require the oxygen. So that's why when you have this type of slant, and you can see this white layer on that. So this is nothing but a strict microorganisms on this agar. So in the case of so when you have, and why we are doing the slant, we have seen that we have talked about in the last class that about the surface area. So generally the slant provides the higher surface area, and because of which the efficient transfer of the oxygen and the efficient transfer of the media components from the agar actually can take place. Okay, so compared to if you are suppose taking a test tube and growing the culture in a vertical way. So in the case of the aerobic system, we require the higher concentration of oxygen. So what we do, we take the agar and we solidify it into this type of tilted structure, and we culture so we streak the cells on the top of it. Okay, so that on the top of it means so now this top surface of this is actually exposed to the air. So generally, that's why for the aerobic cultures, okay, we go for this type of slope culture in which you have the higher surface area, okay, of contact. And in the case of the anaerobic culture, we do not require oxygen for the growth. So what we do, we take, for example, a inoculum loop. So inoculum loop is nothing but it contains some amount of these bacteria colonies or the cells on the end of a end of the loop. So it is nothing but a metallic wire, very thin metallic wire is there, and then contain this metallic wire containing this your microbial isolates. They are inserted within the agar. Okay, and they are placed. So agar, suppose it is a solidified this until this layer. So what is being done? This wire is taken, and with the microbial culture, it is inserted at the bottom. Okay, and then it is kept and cut it from most of the time it is cut it from there, and it's allowed to these microbes are allowed to grow within that. So what happens generally if it is a anaerobic bacteria? So as I told, it doesn't likes the oxygen. Okay, so what is happening in that case, it means the growth of this microbe doesn't takes place on the top surface of this agar. The growth actually takes place within this agar. So in the agar we have some pores or the porous channels that are present. Okay, and within that porous channels, the growth of these microorganisms or the anaerobic microorganisms takes place, and after some time what we can find that like in the initial we can see there is a white cells white layer which is nothing but the microbial culture. So in this case of the anaerobic systems, we can find the sediments, okay, of the cells that are being that is produced at the bottom of the test tubes. So why it is at the bottom of the test tube? Because the oxygen concentration, if we see the oxygen concentration across this gel, obviously the oxygen concentration will be least at the bottom and it will be highest at the top. Okay, because the diffusion of the oxygen across this agar, for example, the agar gel is very low. So that is why if you are going to grow, for example, the anaerobic culture, you have to grow it in the step culture. Okay, so in under the step conditions, means we insert into the microbe in the agar. Okay, and we leave it to grow, and after some time, it actually it grows in number because of its metabolic active state. Okay, and whatever the growth we find after some days, it is below the it is placed at the bottom of this test tube. So then we have seen the other things like we can use, for example, the paraffin oil or this glycerol as a preservant. Like means when you are, for example, have a slant of this microbial of the microbes inoculated onto the surface of it. So we add the mineral oil or the paraffin oil. Okay, and what it helps is it helps in the dehydration of the cells. Okay, and means that's how means generally it leads to the means it inhibits the transfer of the oxygen. Okay, and prevents the dehydration of cells. So when the transfer of oxygen is reduced, the metabolic activity is reduced. Okay, so by that we actually able to preserve the cells for the longer duration time. Similarly goes for the storage in the silica gels. So here what we do is everyone I am sure have seen the silica gels uh, in the electronics packets and so on. So they actually helps to maintain the high, I mean, dehydration. Means it removes the water from the local areas, right? So similarly, when we have means the growth media or containing your high cell density or high concentration of the cells, we add, for example, the finely powdered, sterilized, and cooled silica powder. Okay, and then we store it at a low temperature. So it then it acts like it has the properties of the desiccations that makes it that the cells remains viable for the long period of time. Okay, and then we were so But if you see, means we have studied that the paraffin and for example the glycerol stocks, right, can be used, or the mineral oils can be used for the storage of the cells. But in the last class we have not means seen what is the, means what is the mechanism of action, how these chemicals, the glycerol or the mineral oil or the paraffin oil, they means they are able to prevent or able to help in the preservation. So let us look into that. Okay, what is the basic mechanism? So if we see a particular normal cell, okay, which is at, for example, at the 37 degrees Celsius. So generally, if you see the cells contains means different organelles, right? So it has, depending upon the type of the cells, it has different organelles, and we have different components like the cytoplasm and the cell walls and so on. So in this case, one important things that you need to note is that cells are generally very much means each and every cells have are actually hydrated. So what is the hydrated means? That they have different content of the water in the cells. Okay, and that is a very important parameter to maintain the viability and as well as to maintain the metabolic activity of the cells. So we have seen that the freezing is an efficient way of we can of storing the bacteria. But what if we directly? But we have seen that whenever we are freezing the bacteria, either we are using the glycerol or we are using some sort of additive agents. Okay, and why that additive agents are required? So if we, for example, take a particular this cell, and we go below the below the suppose below the 0 degree. So there are two things that we can do: either we can slowly cool, or for example with the rapidly cool. Okay, so in the both the cases, what will happen in the when there is a rapid cool, what happens? The very small crystals of the ice are being formed, and they actually rupture the cell membranes. Okay, and through this rupturing of these cellular membranes. So in this case, if you are carried out, suppose uh, the this rapid cooling, then generally there is a rupture of the cell membranes that occur because of very small crystals, that ice crystals that has been produced, and this leads to the injury of the cells or the cell death. And simultaneously, if you are going the slow cooling of the ice, in that case what happened, the ice crystals that are produced are generally bigger in shape. Okay, and generally what is happening, there is a solute injury. So what is the solute injury? So whenever, for example, we are doing, for example, any lyophilization or any other process, so what is happening, suppose in your growth media components, what is there are nothing but, for example, some sort of a carbon source. Okay, and there are some sort of a nitrogen source. Okay, and then there are the salts. Okay, so you are for example, you are now freezing it, and then suppose we have seen in the last class you are doing the sublimation to remove the ice. So what is happening, if you see the overall volume of this suspension or the liquid that is there, that is going to decrease. So when the volume is going to decrease or the liquids are going to absent, that what is happening, the concentration of the salts at the local concentration of the salts across the means across these the cells, that is going to increase, because earlier it was what, it was suspended in the liquid. So the concentration of a particular, suppose NaCl as 3 gram per liter, it was dispersed in, suppose, the 10 ml volume of the liquid. But during the sublimation and during the freezing and sublimation, you have removed the liquid. Now the concentration at the surface of the cells for these particular salts, the carbon source, the nitrogen, like maybe the amino acids, and any other solutes. So we have taken the term as a solute. So these actually will be increased at the neighboring surface of the cells. Okay, and their concentration, it might be very high enough that would actually may lead to the rupture, or it may lead to means it may lead to the cell death, because we know that cells cannot tolerate the means high solute concentration. So there is obviously a particular concentration at which we need to add the media components which is suitable for its growth. Okay, so generally this is the challenges that are the two challenges that are there. Like it may rupture the membranes in the case of rapid cooling because of very small formation of very small ice crystals, and in this case in a slow cooling and means if you have the bigger ice crystals and you remove these crystals, for example by lyophilization, and the concentration of the salts, local salts will be higher, that may actually kill the cells. So to prevent these both of these phenomena to occur, what we do is we add the, for example, the mineral oil glycerol, which act as a cryoprotectant. Okay, and what does a cryoprotectant means? Actually, it depresses the freezing point of the bacterial cells, so enhancing the supercooling. So means we have seen that means the glycerol and all this type of oils, they generally actually reduce. So generally the freezing point suppose was earlier at 10 degrees. By addition of the glycerol at different concentrations, generally when we are adding the glycerol, we ideally add at 30%, and we can maximum go to the 50% concentration of the glycerol, depending upon the type of microbes we have, and that actually decreases the overall this freezing point. Okay, and because of that, the formation of these type of crystals are actually are reduced, and that actually prevents the rupturing of the cell membrane. Okay, and if you see that the molecular level, like it is okay that we are able to reduce the freezing point. Okay, and that actually, and because of which we can means we can carry out the cooling of this bacteria at a very very low temperature. Okay, but what is happening at the molecular level? Okay, so at the molecular level, if you see the glycerol structure, so glycerol has a OH groups. So the OH groups are nothing but it's called the hydroxyl groups. Okay, so these hydroxyl groups means this. Can anyone tell what is a hydrogen bond? Have every one of you heard the term hydrogen bond? Yes or no? I'm sure everyone must have heard of it at least in their 10 or so. Can anyone tell what is a hydrogen bond? Harshit, Kirtan.
So it's the electrostatic attraction between the hydrogen, the the hydrogen and the electrons in the oxygen in the H2O molecule.
So in the oxygen is nothing but the electronegative atom, right? So and because of uh, so we have two lone pairs of electrons that are present on this oxygen molecule, oxygen this atom, and what it does that because of that there is a partial negative charge, and whenever there is a surrounding into it, there is the other OH groups. It actually creates a positive charge, partial positive charge on the hydrogen atom, and there is a partial negative charge on the oxygen atom because of these two lone pair of electrons, and because of that there is a presence of hydrogen bonds that occurs. And generally if you see the glycerol molecule structure, okay, so if we draw the glycerol molecule structure, generally they means it has a three hydroxyl groups. Okay, so we are increasing the concentration of these OH groups. So these OH groups, what will happen, these OH groups will actually will form the strong hydrogen bonds with the water molecules that is present within the cell. So as I told you that cells are nothing but they are hydrated in nature. That means the cells have the water inside them. Okay, so whenever we are, for example, adding the glycerol, so glycerol will diffuse, for example, inside inside the cells. Okay, and the overall freezing point is going to be, freezing point of the cells are going to decrease. And why is it going to decrease? Because these glycerol molecules actually will form strong hydrogen bonds with the water molecules that is present within the cells. And because of that presence of this type of enhanced hydrogen bonding, the freezing point is actually depressed, and whatever the effects, whatever the effects like the formation of these type of small ice crystals which lead to the rupture of the cell, so this will be inhibited when the glycerol has permeated from the outside periphery of the cells into the inside cells, and it's reacted with the water molecules to form the hydrogen bonding. Okay, so by presence of this hydrogen bonding, the stability of the cells are actually improved. So this is what is happening at the molecular level. Okay, so and that is why the glycerol and mineral oil and means this oily type of paraffin oil, for example, are used for the preservation of the cells. Okay, so so this is a very basic mechanism why we use the oils and what is going on within the cells. Okay, so let us look into the next topic today, which is on the lyophilization. So we have already talked about that, means what we do in the lyophilization. Like we generally have a lyophilizer, right? Where we maintain a very low temperature. So generally, once suppose you have taken your bacteria solution containing the microbes, you have added, for example, the any of the glycerol or the cryopreservants like the glycerol, mineral oil, paraffin oil, depending upon the type of the microorganisms, then we actually put it into the liquid nitrogen. So here actually what is happening, the ice crystals are actually being formed. Okay, and we make sure that we do it in the presence of some sort of the additives to prevent the rupturing of the cells. Okay, and then what we do is we take these vials into the instrument which is called as a lyophilizer. Okay, and the lyophilizer, we have already discussed what is the basic mechanism by which it works. So at the bottom, for example, you can see it tries to, and there is a column. Okay, and it tries to first of all bring its temperature to, at for example, means at suppose minus 80 degree or Celsius or so. So it is below zero, sub-zero temperature. It tries to bring itself, and then it creates a vacuum. So here you can see a pipe which is connected to a vacuum pump. Okay, and a very high vacuum is being created, and because at that situation, or that means the pressure and the temperature are such optimized so that means the pressure and temperature maintained in such a way that whatever the ice is being there, ice is formed actually during this cooling at in the presence of liquid nitrogen. Okay, that ice should be directly sublimed into the vapors or the gas, for example. Okay, so the ice should not be melting into a liquid and then into the gas. So here in this case, the ice should be directly sublimed into the gas, and by removing those things, we can actually get a very powdered form of the microorganisms. Okay, so this has been the basic mechanism by which the lyophilizations are carried out. And if you see the means the steps that are involved into it, so the first step is the pre-dyeing requirements, like means first of all you need to have, for example, your in the case of the pre-dyeing requirements, you need to have your thick concentrated solutions of your cells, for example. Okay, and in that you are uh, going to add your additives, like we have talked about the glycerol or any other additives. And like for example, you have to prepare what you call the stock, your stock culture containing. So you have the in the stock culture we have the major fraction of the micro high concentrated microorganisms, and rest we add, for example, the glycerol or other mineral oils, which is in the range from the 30% to the 50%. Okay, so these are the pre-dyeing requirements. What are the additives you need to add? Then we have to prepare the ampules, for example, for 20 minutes under under this 20 lb pressure vacuum, and then, for example, we harvest the we harvest the cultures for 3 to 5 days, and we carry out a primary drying. So we have seen that in the earlier case, and like means in the case of suppose the freezing, we do it in the stepwise manner. Suppose you are going to freeze your bacteria up to minus 196. So first we will freeze it at minus 30, and then we will take it to the minus 196, so that means that the cells should not be exposed to any means rapid decrease in temperature. So means if it is immediately put at minus 196 degrees Celsius, then what will happen? The cells membrane will rupture. Okay, and so therefore, in the case of similarly in the case when we are doing this freeze drying of the life or the lyophilization, first we carry out the primary drying. Okay, in which is done for 2 to 4 hours, and almost 90% of the free water that is there in the cell. So free water means the water that is present in the media. Okay, so means we have the cells, and then we on the outside of it we have the media, right? So those media components are dissolved in the water. So first of all, you do the primary drying to remove the water that is present in the surrounding of the cells, which is a part of the media component. And in the case of the secondary drying, okay, it is takes almost 18 to 20 hours, okay, depending upon the moisture content. Whatever the water that is being present inside the cells, that is actually taken out. And in this case, the water is supposed to reduce to below the 1%. Okay, now once you obtain this means these two steps of primary drying and the secondary drying, then you actually, so we generally do these things drying in this type of ampules. Okay, and once you get that ampule, means the dry with the dried culture at the bottom of the test tube, and then we seal these ampules under the vacuum and seal with the flame. So why the flame is required? So flame is required to prevent any sort of the contaminations, so when you are working with that. And then these ampules are then actually stored at a cool room, I mean at a temperature of 4 degrees, or which is in the normal home refrigerators that we there. So we can store it at 4 degrees Celsius for the longer period of time. So whenever you are drying the cultures through this freeze drying method, generally what is happening, the storage time is significantly longer. Okay, so this is the what we means we do, and this is what the steps are in when we are doing the freeze drying of these cells through the lyophilization techniques. Now coming to the other methods, so we have seen these methods which actually so when you are doing this lyophilization, we have seen we are doing it minus 30 and minus 196. So generally, whatever the metabolic activity that is there, that is completely suspended under these conditions. So in this is the methods by which uh, means this is comes under the category of the suspended metabolic state of that microorganism. But there are some microorganisms, as we have already discussed, the microorganisms are very diverse. Okay, so whenever means some microorganisms have a very fragile cell membranes, and they cannot withstand any rapid decrease or increase of the of the temperatures. Okay, otherwise these cells gets ruptured, and overall it is found that the viability of the cells are not there. To overcome that thing and that issue, so we have means a modified method. Okay, so there are general four modified methods, which is like the paper disc, the gelatin disc. Okay, and then we have the pre-dried plugs and the L drying. Okay, so if in the so we'll just look into briefly what are the means the chemical components that are used in this method. So in the case of the paper disc, what we do is we take the thick suspension of the bacteria, and it is placed on the sterile disc of absorbent paper. So we have seen that there are different absorbent paper. Generally, in our labs, we have some sort of a filter paper. Okay, and on that thick suspension of the bacteria is placed. Okay, and means when you are having, we know that whenever means bacteria is actually, for example, placed onto a paper, I mean onto a paper, or and as well as onto an absorbent paper specifically, what will happen? the bacteria that is there, and with the media components that will get adsorbed onto the surface of this type of papers. Okay, and then they are dried over the phosphorus pentoxide in a desiccator under vacuum. Okay, so when we are doing the means of vacuum drying, okay, what will happen? And the phosphorus pentoxide is used because it acts as a means it efficiently removes the water. Okay, so whatever the water, excess water, that is free water. So there are two types of water, right? There are free water and the bound water. So whatever the free water is present, that comes from the growth media, that will be removed, and also the when you are putting the vacuum, the bounded water that is present within the cells will also be removed. Okay, so this is a very simple technique, which is called paper disc method, by which we can suspend the metabolic activity, and then we can. So what will happen, that we will obtain this adsorbent paper containing the only the cells. Okay, and all the media components and all things will be removed. So we just take that papers, and then we store it under the under at a temperature of 4 degrees, and by this way we can actually store these microbial cells. The next method is called the gelatin disc method. So in this, again, a thick suspension of bacteria, like of the bacteria culture, is prepared, and then it is added to a nutrient agar. So gelatin, as the means, I am sure everyone knows, gelatin actually, when we heat it, it is in the liquid state, but at a room temperature, it forms some sort of a jelly-like structure. Okay, and we prepare a nutrient gelatin. Okay, and nutrient gelatin, as the name suggests, there is a some additional components that are there, because that is required, for example, means to maintain the viability of the cells. So in this case, when we are placing a thick suspension of the bacteria on the paper, we are removing the water, but there are some components, media components, that are there, that cannot be removed by vacuum, right? So they are present in the along the surrounding of the paper. So that makes it suitable for the cells to be keeping itself viable. So in this case, since it is a modified method gelatin disc, we add some sort of nutrient gelatin, and the drops of the bacterial suspension in the gelatin are placed on a sterile waxed paper or on a plastic petri dish, and then they are vacuumed. So in this case, we take the nutrient gelatin, we add the bacterial suspensions, okay, and a very small volume of it is either placed in the on the petri plates or on the waxed papers. And what is happening when you are going to apply the vacuum? This particular droplet is going to spread onto this surface of the plates, forming a very thin layer of the microorganisms. Okay, and then we can store these type of plates at a refrigerated conditions. Okay, to store that particular type of bacteria. And the third method actually is called the pre-dried plugs. So in this, what is this plugs? Pre-dried plugs. So plugs are nothing but it's like a cotton wool, which is used, for example, in the first aid or in the dressings. So means in the first aid dressing, and so in this case, we take the pre-dried plugs, okay, and whatever the very thick concentrated suspension of the bacterial culture that is there, we add them onto this sterile cellophane or the pre-dried plugs of the peptone starch or the dextran before drying in desiccator. So what we do is, suppose we have the cotton wool with us, we try to sterilize them. Okay, and then we add on this plugs different components, media components which are required for its means viability. So we have the peptone, starch, or dextran. Okay, and then we dry them into the desiccators. So desiccators are nothing but a closed chamber where, for example, the means we also have a dehydrating agents that are present within that closed chamber. So once we have, for example, these pre-dyed plugs, okay, containing the sterilized plugs, and then we dip it in, for example, into this solution, and we dry them out, okay, and then after that we add the microwave culture, and then we carry out the vacuum based drying. Right. So, what will happen? This whatever the microbes are there, they will be means incorporated onto this type of woolly substances. Okay, and then it can be stored at that for the longer duration of time. The next and the last technique is the L drying technique. So, it means it is used, for example, for the microorganisms which are very fragile, and it's very difficult to work with them. So in this case, what is there? Bacteria in the ampules are dried from the liquid state using a vacuum and desiccant and a water bath to control the temperature. So it is a very simple technique. Everyone knows that we know, like the water boils at, for example, at 100 degrees at a particular atmospheric pressure. That temperature will decrease if we induce, if we, for example, add the, for example, vacuum pump to it. So, for example, if you we can, so at this temperature we know the water starts to boil. Okay, we can reduce the temperature, and for example, we can reduce it to 40 degrees Celsius, and we can create a vacuum pressure of, for example, the 555 megapascal. What will happen? At this case, at a 40 degrees Celsius, the water will start boiling. Okay, because of the lowering in the pressure. So what we do is, generally we take this growth media components. Means say we take the thick suspension of this bacterial culture, and we put into a water bath. So what does the water bath does? So generally it is something like this, where we have with the suspension, and then we place it onto the water bath, which is having a higher temperature, suppose at 37 or 40 degrees Celsius, high enough that it doesn't kill the cell. And then we try to maintain means we apply the vacuum, and when you apply the vacuum, what is happens, the water starts to boils, and it is being converted into the vapor. Okay, and the vapor is removed and condensed, and what we leave behind is the microorganisms. Okay, so this is the fourth technique of the L drying technique, and so these are the four ways by which we can actually preserve the strain. And whenever you are, so we have seen that the preservation of the stains required the subculturing of the cells. So it is generally ideally suggested not to transfer more than the 5 passages from the mother culture. So mother culture is the first culture that you obtain. Suppose when you are taking the freeze dried, this type of, generally when we work in our labs, we order this type of freeze dried cellular samples. Okay, and then we start with the first culture by making the some of the subcultures. Okay, so that it becomes what is called the mother culture. Okay, and then that mother culture is being subcultured for several cycles. Okay, and a small fraction of this culture is being taken and is being preserved in our database. Okay, so it should be there that when you are passing these cells, you are taking a small volume, putting into another, taking a small volume, putting in another. So during that, it should not exceed the 5 passages. Okay, because this is a standard, some standard type of thing, you can say, because if you pass the cells for longer duration of time, there is a higher possibility for some genetic or the phenotypic change. So just to prevent that, we do not ideally transfer over the 5 times. Okay. So let us. So this is comes to the ends of the topic of this preservation of what are techniques, and what is the how we can preserve the industrial stains. What is the very basic mechanism? Suppose you are doing so you are doing with the aerobic anaerobic, and then in the case of solid media, we have two options like the slope culture and the step culture, and the other methods of the freeze drying, importance of the glycerols, and also so on. So let us start with the next topic on in the next class on like how we can what are strategies to improve the strain like the mutation, the portable solution, the different recombinant DNA technology, genetic engineering, and so on. So we'll look into that in the next class. So anyone has any questions or doubts until now? What you have studied? Anyone? Hello? Hello?
Yes, sir.
Yes, sir.
Sure.
So if no one has no questions or doubts, so let us. So thank you everyone, and let us meet in the next week and continue our discussion for.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir.